you ever wanted to know about the production of biofuels?
Not really.
Okay, how about testing for your ancestry through DNA?
Doesn't sound too interesting.
What about a revolutionary new method of splicing genes in order to make organisms better suited for fighting diseases and conditions?
Sounds interesting to me.
Genome editing is a process where DNA is inserted, replaced, or deleted in the genome of an organism using engineered nucleases or molecular scissors.
An explanation will be provided on the basic information, the history, how it works, as well as present and future applications.
Without further delay,
let's go.
To start things off, the discovery of DNA happened in the late 1860s by Friedrich Miescher, a physician and biologist.
It actually took 143 years for scientists to be able to accurately map the human genome. They finally did it in 2003.
In 2007, the TALEN method of gene editing was introduced, revolutionizing how gene repairing was accomplished.
In fact, the TALEN method was even voted as the best method of 2011 by a famous organization called Nature Methods.
In April of 2013, the new CRISPR/Cas9 method, which we'll be referring to as CRISPR for the majority of this video, was tested on a zebrafish. It only took until June of that year to make CRISPR totally friendly for scientists.
In 2014, the CRISPR method was enhanced even more to allow precise gene editing.
The technology of CRISPR now holds the spot for what scientists are really focusing on, versus the TALEN method.
Unless they are looking to treat a disease caused by a mutation in an organism's mitochondria.
Oh yeah, almost forgot about that one. Yes, diseases can be caused by mutations in an organism's mitochondria.
As I was alluding to previously, the method of gene editing that scientists use to treat mitochondrial impairments is indeed TALEN.
What are TALENs?
What? Dude, you already know. Oh, glad you asked. TALEN is a way to cut, repair, and edit strands of DNA.
Okay, then how do they work?
The TALEN compound is separated into two different, yet essential parts. The TAL component is used to locate the exact gene section in a cell that is causing the disease. The EN section cuts the broken gene into pieces. The fresh gene copy is put in place of the broken one, and it's off to the races. Homologous recombination is used to rearrange...
Whoa.
What?
You gotta tell them what it is first.
Oh, right. Homologous recombination is a natural process used by cells to repair simple breaks in DNA strands. It's used to rearrange the new gene's DNA letters so it works properly with the pre-existing strand.
Oh, that's what that is. Sounds a bit like CRISPR to me.
What's CRISPR?
CRISPR is a newer method of gene editing. It accomplishes the same task as TALEN, but is a little more precise.
So, how is it different?
Glad you asked. CRISPR is also split into two sections like TALEN, an enzyme and a guide. The enzyme is called Cas9, which does the job of cutting and repairing the DNA. The guide is a piece of RNA called guide RNA, or gRNA. This RNA helps the enzyme find the right section of DNA to cut.
Cool. So the CRISPR method and TALEN method are both good ways of getting the same task done.
Yep, and CRISPR will even keep a piece of the virus behind to help with future attacks from the same virus.
Gene editing has quite a few important purposes.
For example, human genes are being edited as we speak to help treat diseases and disorders.
A technique this new is bound to have possible consequences. For example, cells could turn cancerous if wrongly modified.
Of course. Another method that is highly controversial is the modifying of human embryos to help with diseases and disorders before the human is born.
Obviously, this is a debated topic and a bit unethical. However, many scientists have actually gotten the green light to go ahead and delve into this project.
Finally, here's a different spin on the gene editing methods. Instead of human genes, lots of research and experimentation has gone into modifying the genes of farm crops.
Crops like corn, barley, and rice can and are being modified to make them hardier against climate and easier to grow.
All in all, gene editing is being utilized right this moment and will have many more uses in the future.
And that's about it.
We left out a few specific details, but that's a brief explanation of gene editing.
First, we started out with a brief history on gene editing.
Then we provided a little bit of information on mitochondrial diseases.
We also explained how the TALEN and CRISPR gene editing methods work.
Yep, and we finally wrapped up with present and future purposes of gene editing.
Well, that's all for this video.
We hope you enjoyed,
and we'll see you guys
in the next one.